if you are heading the department like clinical microbiology and you are going for the daily round, then what you should assess from the technicians and from the uh, doctors there. So I tried from ChatGPT and it is from there. So as head of clinical microbiology, your daily round should assess not only technical quality but also patient safety, turnaround time, infection control and compliance with the accreditation standards. So daily HOD checklist for bacteriology lab. As you know, in my lab we have 7 labs: bacteriology, serology, molecular lab, TB lab, virology, and micromolecular is one and the same lab. Then we have TB lab, DMC lab. So these are the various labs we have in one microbiology lab. So when we go to bacteriology lab, what all we should check, like sample receiving. First of all, you should see that are all samples properly labeled with patient identifiers, are specimen acceptance or rejection criteria being followed, are any high priority samples pending, is specimen transport time was appropriate. So this all you have to check when you go to lab for sample receiving. This we can learn in detail also. Second thing, from after sample receiving, the first thing we do in microbiology is direct microscopy. That is, directly we see what is there in the sample. So are gram stains being performed correctly? This you have to see whether control is there or not. Is gram stain quality satisfactory? Is gram stain correlated with the culture findings? Are critical gram stain findings immediately communicated to clinician? This is important regarding direct microscopy. So many things are there, but as far as the round is concerned, you cannot go for it for whole day. So for daily round, this much is important. Then coming to the culture processing, correct media selected for each specimen. So if urine is being processed on clad, and chocolate agar is there for CSF or not, these type of things can be checked. Incubators functioning at correct temperature, because after processing one has to incubate. So condition of the incubator, plates labeled correctly, anaerobic cultures processed appropriately if applicable. So when you are seeing plates labeled correctly, all the date, the sample, and how labeling is done in your lab, that you have to check. Anaerobic cultures processed appropriately if you are doing it. Then go for identification, significant pathogens identified correctly or not. If there if you find any pathogens, then biochemicals and all that. If you are not doing automation, then you have to see what biochemicals have been put for it. Mixed growth interpreted appropriately or not, because that is very very important. How many colonies you have isolated and how many are there, which one is pathogen, which one is contaminant, this you have to find out. So contaminants differentiated from pathogen or not in this case. Repeat cultures advised wherever indicated. So if was it done or not. So these are the things on the next step what you have to check. Then is the antimicrobial susceptibility testing. CLSI guideline followed or not in case of antimicrobial susceptibility testing. Appropriate antibiotic panel was selected or not. If the VITEC is being done, or if the automation is happening in your lab, then internal quality control, strains acceptable. So MDR, XDR, PDR organisms identified. If yes, then they should be properly record should be maintained, and it should be reported to the HIC team. Unusual resistance pattern verified. Reporting is the next point. So, critical values communicated and documented, reports clinically relevant and complete, turnaround time maintained or not, pending reports reviewed or not, or if anything wrong, or if wrong reporting has been done, then whether it is corrected or not. So, quality management is NABL. IQC record should be checked. Equipment calibrations are up to date or not. Then temperature logs are maintained or not, because that can affect the result. Non conformities were documented or not. Corrective actions implemented or not. So these are regarding the quality management, that internal quality control should be there, equipment should be completely calibrated, temperature log should be there, because this all is going to affect your results. If any non conformity is there, then there should be a record for it, and then what corrective actions were taken, that should be maintained. So, antimicrobial stewardship, any carbapenium or colistin resistance detected. So, this is important. So, one has to keep an eye on it. If any ESBL, MRSA, VRE, or CRE isolates are there, that is also very important. Why it is important, this we will discuss in the next video. Then, clinicians were informed or not, because only then they will change the treatment. So, that is important. Then stewardship interventions documented or not, this is also very very important, that what how you report to the team and how the action was taken. So next is infection control. What all you are going to check here, that any alert organism reported promptly or not. This is very very important, because if any resistant organism has been identified, then you have to inform it immediately. ICU and ICU isolates reviewed or not, any outbreak signal, so you should take care of it, that how many samples have been reported from ICU and ICU. These are the places where special attention should be given for, because serious patients are there. So what is happening in their reports, that is very very important. So it is better that the people who are coming from ICU and this, and how many from wards, and how many from OPDs, so we should try to see according to this thing also. Then environmental cultures pending or not. Then in the teaching, whatever what all is going on, because this is medical college, so every time we should discuss one clinical case with the resident and ask for gram stain interpretation, ask one AST interpretation. This is also done. So you can ask your PG with the report, you can ask the SR with the report. If you are not able to go for the discussion properly at that particular time, then you can discuss with them. Review one difficult culture. If anything has come and it is different, then that can be discussed too. Then what we should ask with the technician. So in regard to specimen handling, specimens were received correctly, proper labeling verified, rejection criteria followed or not. So we should keep asking them, we should keep giving them training, and if keep asking some questions with them. Then media quality were checked or not, quality controls were put or not. So expiry dates verified, did he do that? Then sterility records were maintained or not for the media. So equipment is incubators were checked or not, proper cleaning, proper temperature should be maintained, refrigerator temperature should be recorded, biosafety cabinet disinfected or not, and whether they are functioning or not functioning properly. But for technician, he should see ki whether he checked the temperature in the morning, whether he checked the temperature of the refrigerator in the morning. This will show that how the this equipment has worked overnight, and now also it is performing in the right direction. So everything was kept on the right temperature. Then biosafety cabinet was disinfected or not, it is very important. Autoclave is functioning or not. So these are the important equipments, and these equipment should be discussed with the technicians who all are taking care of it. And we will try to provide the uh, things in detail, that what ideally it should be asked with regarding to that. Then coming to the culture work, then what you should ask the clinician, that correct inoculation technique was used or not. They should have the knowledge of the inoculation techniques. Then plates labeled properly. So many times you will see that they are not able to isolate the colonies. So that is very very important, and one colony is there and other is under it. So this is not okay. A colony should be properly isolated. Plates labeled properly. Incubation condition should be correct. So this is regarding the culture work which you are going to ask with the technician. Performing gram stain is again a good technique and expertise. So reagent should be fresh. There should be no crystals and no deposits. Smear quality should be satisfactory. Microscope should be cleaned. With what thing they are cleaning the microscopes, so these all things should be asked with the technicians who all are there. They will tell you. After that, regarding AST point 0.5 McFarland standard, prepared correctly, antibiotic disc stored properly, disc expiry should be checked, quality stain performed. Documentation, bench worksheet completed or not, TAT is maintained or not. Then critical results informed to the microbiologist. Coming to the biosafety, was the technician wearing the PPE? Does he has knowledge about PPE or not? Then biomedical waste segregated correctly or not? So biomedical waste should be checked. PPE for technician, workbench disinfection, how it was done, and hand hygiene was followed or not. Proper steps of hand hygiene should be followed. At the end of the day, pending cultures were handed over to the other person. So proper give and take of samples should be performed. Incubators should be checked at the end of the day. Equipment switched off on as required, and laboratory should also be cleaned. But if it is 4 hours, then it should be the other technician should know each and everything regarding it. Then questions to ask during your round. um, which samples are pending beyond expected TET and why? Any positive cultures today? So blood cultures are important. For MDR organisms, how many MDR were there in the lab? Any unexpected antibiogram requiring the repeat testing? Any rejected samples and their reasons? Any equipment or reagent issues? Any alert organisms requiring infection control notification? You should go and you should alert the clinician. Which ICU patients have significant culture results today? Were all critical values communicated and documented? What quality issues was identified today? So this is the checklist which is suitable for 15 to 20 minutes of round, because you cannot ask each and everything, and it is, I think, I don't think so many things this has covered, and this has covered so many things in this video. I think if we are going to take it as a 15 to 20 minute round, it is more than sufficient and a good checklist. But you can also add something if that is required, or if it is mm, according to your labs, align well with the laboratory quality practices, antimicrobial stewardship, and this thing. So this is all about the this is all about the general round. And that I think that yes, it has covered many points. So if you want, you can make the list and you can keep it in your lab so that you are not going to forget. This is a good thing. Or gradually you can add other things too, but this much is because when you make a list now, you never forget anything. It is also not very important for all the people, or all days. But most of the time, if you are trying to cover it, if you give the duty to the microbiologist that these all points you have to tell me when I come for the round. So if you are going to forget something, they will tell you, they will help you that yes, these are the things. And I think if you make checklist, then you never forget anything, and you can go serial wise, not means not by skipping anything important. So this is very good. If you like it, you subscribe it if it is helpful for you. Otherwise, I just go through all these things just for the revision sake, and to see what ChatGPT can um, give you and how it can help you. So thank you very much. We will discuss the sample collection in the next video.